So with that, we've got Jordan Bryan here next, who's a professor here at UVA in the School of Data Science. And I had one conversation with him where I realized there's actually a remarkable amount of potential overlap between the things that he's been thinking about in terms of spectral unmixing. And so with that, Jordan, I'll let you take it away.
Yeah, great. Thanks for having me, everybody. And I'm glad that the two talks have so much overlap. So I think a lot of just, our brains are now thinking about spectra. So some of the intro, maybe we can go over a little more quickly. I'm going to talk about a particular estimation problem in the context of unmixing spectra. So where we're interested in a specific estimand, like a quantity that we want to estimate. I encountered this problem in the context of water quality monitoring using fluorescence spectroscopy. So like a particular technology in a particular setting, but maybe if you guys can, see the analogies to physical settings, then that might be, or astronomical settings, that might be helpful and productive. So let me start by just talking about kind of a core workflow in statistics. This is what statisticians often think about. One posits a statistical model or some set of models that describe data that you might see. You propose a class of procedures that use this data because you want to get at some underlying quantity. You define some optimality criterion by which you can assess your different procedures. You then use this optimality criterion to find an optimal procedure for mashing up your data to arrive at some number. And then sometimes it's the case that you can actually do what's optimal. More often than not, what you have to do is then find some kind of computable or feasible approximation to this optimal procedure that you found. All right, so today I'm going to talk about a particular instance of that workflow in the context of this spectral unmixing problem. We're going to talk about a classic type of estimation. This is linear unbiased estimation. Typical optimality criterion there is something called the mean squared error of your estimate. In this case, because we have unbiased estimates, we have the mean squared error is just the same as looking at the variance of your estimator. The optimal procedure in this class is something called the best linear unbiased estimate. So that's the blue. It makes sense. And then so really kind of the main takeaway here is we're going to propose one feasible approximation to that best linear unbiased estimate. It's very simple to compute, and it also kind of indicates I think something interesting about how one should use a collection of spectra if you're still lucky is to have them. Okay, so just what do we mean by variance? I just want to be very specific about what that means because I know that the audience is mixed here. What I'm plotting is on the left, you have some relationship between, let's say, the concentration of some chemical and then some emission intensity or the log of that when you've measured this substance with the spectrometer or something like that. You can imagine this is just at one peak location. So as the concentration increases, so does the emission intensity. And one could say, let's just assume that the variability around the central trend here is constant for the different values of the concentration. If I do that and then I estimate the slope of this line using least squares, on the right is a histogram of the values of that slope that I'm going to get if I kind of run this experiment over and over again. So we get the black line is the true slope on the right, and then we get some spread around that because when we run the experiment over and over again, we're going to get different estimates of what that thing is. Okay, well, the data that I used, or the model according to which I simulated the data on the left is actually not this constant variance one. It's a model that has something called heteroscedasticity. So it has increasing variance of the emission intensity as you increase the concentration. And more or less, this is actually how things work with at least fluorescence spectroscopy. And if I find the optimal estimator in that model where I don't assume that the variance is constant, if I know the pattern of increasing variability, then I can propose a different kind of least squares estimate that takes advantage of that variability. And what you get, so variability around the central trend, and what you get in terms of the variability of the estimate is less. Okay, so here we're estimating using basic least squares where we've ignored this fact that the variance is increasing as the concentration increases. Here we're using something called generalized least squares that uses that information, and we achieve something that's, again, unbiased, so it's centered around the truth, but we have a tighter spread. Okay, so that's what we mean by the variance of an estimate. And if you look at these plots long enough, you'd say, okay, I'd probably prefer the red one because on average, the number that I get is going to be closer to the truth. All right, so that's linear unbiased estimation and optimality. So what's the overall problem? This talk was better when I was at Duke and UNC because the motivation was super close to home, but I think North Carolina is close enough to Virginia to say that maybe we still care about river systems in our neighboring state. So I worked with some folks from NC State University on a project where people in the Oceanic and Atmospheric Sciences department wanted to measure the water quality of this Neuse River. And you can imagine that they took the samples that they're ultimately interested in are here kind of furthest downstream. And the circles along the way are illustrating different types of potential polluting sites where the water would flow, water from these polluting sites would flow into the river and then show up in some sort of chemical signature measurement downstream. So you can imagine that like further up towards Raleigh, there might be like street runoff water that has its own chemical signature. There could be what we'll call a swine lagoon. So just runoff water from where pigs live in Clayton, or maybe just another farm without livestock, but where you have a lot of crops or something in Kinston. These all have kind of unique chemical signatures in terms of the water that runs off from them into the river system. We'll call all that stuff dissolved organic material. All right, and we quantify that dissolved organic material using fluorescence spectroscopy. I think luckily maybe for the physics audience, I can just say that that's what they use and then move on instead of trying to pretend like I know anything about it. So you guys know more about that than I do. Let's look at some spectra. On the left, we have a soil sample. So this is an actual spectrum that was collected by these researchers from an agricultural site. And on the right, we have something from one of these swine lagoons. And just kind of at a very quick examination, you can see that these two spectra have different shapes. The kind of the mode is different between them. There are two modes for the soil sample in particular. So one would think that maybe if you looked at these, if you had other examples from these two sources, you could disambiguate the two just by looking at the shape. All right, so the data that we got from these researchers wasn't just some individual spectra from each of these kind of pollution contributing sources. We got many. So we were sampling, in a sense, from some distribution of spectra. In this case, we'll think about them as big letter K, different polluting sources. So one of those sources is from the farm, and one of them is from the swine lagoon. And you can see that there is some sort of average fluorescence profile going on here for both, but then there's also some variability around the average of the fluorescence profile. So I'm sure that not everybody does the same thing, but at least it was my experience that oftentimes when people try to solve an unmixing problem, they'll use some kind of like best representative spectra from each of many categories and then do some kind of least squares fit. What we're going to talk about today is a different way to set up that unmixing problem where we don't choose a best representative spectrum. We actually use all of these spectra to get a better estimate. All right, so what is the estimation problem specifically? We have this downstream spectrum measurement that we're imagining is taken from the river, furthest downstream from these polluting sources. And we suppose that it is a linear superposition of the constituent spectra, and that linear combination is weighted by some weights, theta 1 through theta k. All right, and that accords with, again, something from physics called Beer's law, which I will not try to explain to you guys, but this kind of allows us to assume an additive model in the world of statistics and say that that's somewhat realistic in reality. Okay, so we observe this purple line, and we want to estimate the abundances, theta 1 through theta k, the weights of this linear superposition. All right, so what makes this problem a little bit different than just the most standard linear regression problem? You can imagine it's really this notion that what we see is the downstream fluorescence profile Y, but the the summands in this linear combination are not observed themselves. Okay, so the key there is that when we measure a downstream fluorescence profile and we assume that it's a sum of individual spectra, we can't take the downstream water sample and physically separate it into the separate parts and then measure those spectra. We don't have those. Instead, what we have is we have these samples that I talked about. So these are instances of spectra from each of the polluting sources that we actually observe and measure, but it's not the one, it's not the exact constituent spectrum that went into whatever the downstream sample we observed is. So sometimes in statistics, that's called an error in variables model. So we're not going to get into like a lot of the specifics of that here, but just to introduce that terminology in case anybody's familiar with it. To repeat, we have n sub k, so some number of observed instances of spectra from different polluting categories. And then we have this one unobserved one, and we're assuming that these are independent and identically distributed from some unknown distribution of spectra, one for each of the polluting source categories. All right, so in the next set of notations, I just want to note that what we do is we assemble all of the observed profiles. So we have n sub k from each source category. We're going to have little n as the total number of profiles. And we're going to take all of those and we're going to form what we call a dictionary of spectra. You can imagine the wavelength of the spectra being the rows of this matrix, and then the cell intensities in this diagram being like the height of the emission intensity here, or the fluorescence intensity. For our purposes, the fluorescence profiles are actually, we have several emission spectra for several excitation wavelengths. So everything's a little bit more high dimensional than shown. So the number of examples we have is actually less than the dimension p. So this matrix is a little bit misleading. We have a longer matrix than it is wide. All right, so two kind of computationally very cheap and simple methods to try to estimate these abundances theta. One we call the average then regress method, because that's what we do. What we do is we say, let's assume that this downstream fluorescence spectrum is roughly a linear combination of the average fluorescence profile from each source category. Seems reasonable. We average, and then we do regression. Regression means computing this least squares solution in this case. We get the average then regress estimate. Another estimate we call the regress then sum estimate. All right, so this one says, let's say that the downstream profile is actually sitting somewhere near the column space of all of our spectra. So not just in the column space at the averages, but some linear combination of all of the spectra. Now we have a vector. If we do least squares and solve for this beta, we have a vector that's n long, and we need something that's k long to get one number for each of the source categories. To get our estimate, we do this least squares regression. That's this part of the equation. And then this A matrix is taking the sum of the regression coefficients within each source category. This is just a matrix of zeros and ones that's just taking the sum of the regression coefficients that you get within each source category. Okay, so you do regression and then you sum. That's RTS.
Can I interrupt?
Yeah.
The difference is that you have the averaging in the first time, you have it in X, right?
Yeah.
Right. And that means that it's in the inverse, which is
Yes.
the only reason why this is nonlinear. So why these are different is because it appears in the inverse.
Yes, that's right. Exactly. Yeah, exactly. You can write this X bar in terms of the A matrix actually, and yeah, exactly. Great. So quick poll. Who thinks that average then regress is better?
Oh goodness.
There's nobody. Who thinks that regress then sum is better? More people. Okay, so people, it's good. Pattern recognition. Usually the second one is the better thing. Exactly. So everybody is right, but let's think about. So I'll explain why. I'll explain why in a little bit of detail. So rewriting the sampling model that we already had, we have the downstream profile is a linear combination of these unobserved profiles plus, potentially some noise that I'm representing with this zeta term. If that's the case, then on average, this unknown profile we'll call this matrix of true average profiles. We'll label that with M. And we're going to assume here that the variance of both this additional error term and the variance of the individual unobserved profiles all are just some common covariance matrix sigma. All right, so if that's the case, then what our model is saying is that on average, our downstream profile will be a linear combination of the true, these kind of population level average profiles, and the variance is going to be proportional to that common covariance matrix. All right, so if we knew the M and the sigma, then we could obtain what I'll call these oracle meaning we know these true quantities, which we don't actually have access to. But if we did, then here are some oracle linear unbiased estimates. One is the ordinary least squares estimate, which uses the matrix of fluorescence profile means and just solves the least squares problem. The other one is called the generalized least squares estimate, which uses both these true means and then also this true error covariance matrix. All right, so in general, it's the case that this GLS estimate is like that first example that I was showing. It's the generalized least squares estimate. This is the best that you can do in the general case where this covariance matrix is not proportional to the identity. Right, so you don't have, it's not just constant in that one-dimensional example, but it's also, it's not constant in, all the directions like the identity describes. All right, so the key observation that we made about spectral data is that the error structure is it's not isotropic. So the actual answer to which of these two estimates performs better, the real answer is it depends, because that's always the answer. But with spectral data, it's the case that the error structure just will have structure. It's not going to be this kind of perfectly unstructured independent error. Okay, so when that's the case, we can realize significant gains in efficiency by using the generalized least squares estimate instead of using the ordinary least squares estimate. All right, so we don't know these things, this M and the sigma, but we can obtain what I would call a feasible substitute for both by estimating those things using our dictionary of fluorescence profiles. And without going into the details, here's the punchline from the article that I wrote with my PhD advisor. A feasible version of this ordinary least squares estimate is the average then regress estimate. I think that that's not too hard to see. You take the profile averages to estimate the M matrix, and then that's what you get. A little bit more linear algebra and moving things around shows that a feasible generalized least squares estimate, where you find plugins both for this average average fluorescence profile and then also the error covariance matrix, that is actually equivalent to this regress than sum estimate. Okay, so two very simple procedures, but we can use the theory of linear models and optimal estimation within the context of linear unbiased estimators to show that this regress than sum procedure approximates something that is kind of optimal in the general case. And then empirically, we've observed that it does a lot better than this average then regress procedure. Okay, so one take home from this is that if you have a whole collection of spectra, i.e. many samples from representing some particular category, you shouldn't just pick a best representative or just use the average and then do least squares. There are better things that you can do with all that information to get a more precise estimate of these abundances. All right, so our collaborator from NC State put this to use, and these are the kinds of things that you can do. You can look at water samples collected over time near a particular river. And in the bottom plot, what's shown is that there's this is precipitation in inches. So you see a rain event occur, right after which there's a spike in this contribution from the swine polluting category. Okay, and this was taken kind of near one of these swine lagoons, or at least a river that is close to one of these polluting sources. So that's nice to see that you can see things occurring over time. I will say that nothing, these quantities were estimated without taking into account the time structure. Like the actual estimates were just computed on the profiles themselves. So yeah, there seems to be some real signal there. So yeah, so some things that I'm thinking about in relation to this, and I mean, I hope potentially in relation to some applications in astronomy. It's the case that if you don't assume this common error covariance matrix, you end up with a little bit more of a complicated model where the variance of that downstream fluorescence profile depends on those weights as well as the mean. So you get this like kind of quadratic structure in there. You can show that the amount of information kind of from that model is not being fully taken advantage of with even the best linear unbiased estimates. So if you look at other types of estimates, you should be able to do even better. I think especially in regimes with like low abundances, you can estimate those more precisely. Another thing I'm thinking about is what I would tell a practitioner to do is don't do anything that actually I set up here exactly. Just find non-negative versions of these estimates. Empirically, you can show that a non-negative version of this regress than sum works better than a non-negative version of the average than regress. But precise non-asymptotic theory of these kinds of constrained estimators is yeah, it's definitely a lot more challenging than just like using linear model theory. And one could take maybe a Bayesian approach and get that kind of thing, but certainly from the more classical statistics perspective, these constrained estimators are harder to analyze. So that's something I'm interested in. And on a related subject, getting some kind of again, kind of a classical statistical notion of the uncertainty in those estimates is also really challenging when you move to the non-negative setting. And this errors and variables framework as well, that I mentioned kind of at the beginning of the talk, also introduces all kinds of other difficulties in getting good error bars on those thetas. And then finally, this is less of a statistical problem, I think, than a problem that would be best approached with domain experts. But what we're getting is some kind of unitless relative abundance. Hard to say exactly what these numbers mean other than relative to the other coefficients that you're estimating. If you're interested in getting absolute numbers and you want to say something like the concentration of this substance in this sample is, precisely this with some error bar, that takes a lot of work dealing with the quirks of particular instrumentation, and then also like probably would differ greatly between applications in astronomy versus applications in environmental monitoring. So those issues, if you're interested in absolute abundance, take a lot of work kind of on the front end. And yeah, maybe less my domain, but certainly of interest to me. And I think in particular, we already saw a bunch of pictures of spectra, so I think that again, I guess I don't have to convince you that there are applications for this sort of thing in astronomy, but it does strike me that the particular problem that I've been considering might be of a little bit of a different flavor than what typically goes on, and I wonder whether there are places where as opposed to knowing for sure that this element causes this exact peak, you might have more like, that a group of spectra belong to some kind of loosely labeled category, but it's a more complicated substance, and yet still you'd like to sort of estimate how abundant is this, kind of more ambiguous thing in some other spectrum or some other chemical mixture. So yeah, I'd be super curious to hear whether there are analogies that occur to anybody. So with that, I'll finish up, I think, before 1. So if we're still here.
Thank you, Jordan. And yeah, as I mentioned, at 1 o'clock, the room will like auto disconnect. And so I wanted to prioritize if there's any questions from folks online. I'd be happy to leave space for that first before coming to folks in the room here. Seeing none. There's 2 hands up. Got it. Stephanie. This time
Paul, can I
please.
ask a question real quick, Paul? Sorry, I can't raise our hands in the room here very easily. I was wondering, so here you kind of assumptions about linear mixing, but one thing that we often encounter in astronomical context is nonlinear combinations due to radiative transfer. And so I was wondering how that kind of fits into this bigger picture. Is it just completely irrelevant in a different problem, or are there ways that you can tweak this for sort of nonlinear mixing?
Yeah, I think that one should be able to tweak it. So I guess maybe the first thing that comes to mind is that at least, so if you have some nonlinear model and you were to do some kind of maximum likelihood estimator or something like that, there are at least large sample approximations that you can make where you can get something close to what the distribution of that estimate should be. And actually also, if there is some kind of linear component or like additive error component where you you know, if the error structure is somewhat additive, or at least you can approximate it as such, then some of this generalized least squares framework still carries over. It just becomes, they become large sample results as opposed to things that you can say like this estimate has this distribution exactly. But I think they would certainly be carryovers. It would just be more work involved.
Got it. Thanks.
Stephanie, I saw you had your hands up. Glad to get to you. We're at the sort of 90-second mark. So quick question, quick answer.
It was answered all in the chat, so I think we're good.
Ah, okay. Good. Yeah. So just to note, I see a lot of parallels for quasar absorption line spectroscopy. I mean, I would actually think about the problem differently, and I'm glad to unpack that with you. I would have thought about it as being additive in optical depth space.
Sure.
Because we're thinking about passing through more and more stuff. And anyway, be really curious to explore that a little bit more.
Awesome.
There's also, I would say, in planetary science, you'd find that too. There's, for instance, CRISM is a hyperspectral imager on board of the Mars orbiter. So you can see basically planetary surfaces, and the composition of the reflected light is not unique. So you can have the same mineral composition, but you see it on the different angles, and you can get a different response. So I think that fits quite nicely to what you said earlier.
Thank you. Yeah, I was also curious from the planetary folks about secondary transit analysis. But I think with that, I want to take these last couple of seconds to thank everyone for coming once again, especially on this busy proposal day. Thank both of our speakers for these great talks, and look forward to seeing you guys in 2 weeks. Keep an eye out for that announcement, which will be happening not here at UVA, but I think at one of our partner sites. So thanks again to our speakers.